really going to be about how viruses evolve, and this was a subject that my lab has not so much worked on, although I'll present a little data from my lab that shows how we became interested in this phenomenon. And it was a topic that my colleague, Dr. Galasho, had suggested might be of interest to this group. So um, I think everybody can appreciate the idea, and this is sort of dogmatic, that viruses evolve rapidly in the short term. So probably the best data that supports this is from the millions of sequences that have been generated for SARS-CoV-2. And with all those millions of sequences, you can generate plots like this that show that SARS-CoV-2 is evolving rapidly at a steady rate of about one mutation per 1,000 bases of its genome per year. And this is pretty linear evolution over time. So over 5 years of the pandemic, the SARS-CoV-2 genome has accumulated about 150 mutations. And so that corresponds to an evolutionary rate of about 10 to the minus 3 substitutions per site of the genome per year. And that's the units when we talk about how fast viruses evolve. So it's clear that viruses evolve rapidly in the short term, but evolution is a process that plays out over long time periods as well. So what happens over longer time periods, and how would you even know, where would you even get long-term rate estimates for how fast viruses evolve? Well, one straightforward way to get information about longer term virus evolution is to sequence viruses from old samples. And this is one of the ways that I became personally interested in this phenomenon and this issue is because a student in my lab, Lexi, had been sequencing RNA viruses from old entomological museum specimens. So on the left of the slide, you can see some of the old insect specimens from museums that she extracted RNA from and sequenced. And this bug on the top is actually from 1899. And she recovered RNA from these sequences, and I'm not going to really talk about that project, but it's a really cool project, and there's a preprint if you'd like to read more about it. But you can directly sequence viruses from old samples, and then compare those sequences to current contemporary sequences to infer how fast the viruses have been evolving. And that's kind of what's shown in this tree on this slide, which is showing old sequences, which are shown in purple, in the context of contemporary sequences, which are shown in black. The other way you can get really even longer term rate estimates is to look at these endogenized viral elements. So these are also called the EVEs, endogenous viral elements. And these represent historic integration events of virus sequences. And this can occur for DNA or for RNA viruses, where these sequences occasionally, rarely, but sometimes get captured in the sequences of eukaryotic genomes. And when this happens, sometimes those genomic sequences can go to fixation in the population, and they can even show up later, far into the future, in the sequences of multiple related species. Then you can infer when the endogenous sequence integrated into the genome, if that integration event is preserved at a syntenic site in the genome, and then you can look at when those two species diverged to infer when that integration event occurred. And an important detail is that once those integrations occur, that sequence, which is now genomic, only evolves at the rate that host genomes evolve, which is much slower than virus sequences evolve. So if SARS-CoV-2 is evolving at a rate of 10 to the minus 3 substitutions per site per year, our genomes, the genomes of eukaryotes, are evolving 1 million times slower at a rate of 10 to the minus 9th substitutions per site per year. So endogenous viral sequences are another way to capture old virus sequences. And this is a project that I was involved with a few years ago, where we characterized some new snake viruses that were infecting these carpet pythons in Australia. And these are Bornaviruses, a type of negative sense RNA virus. And on this tree on the right, you can see the red arrows point to the two snake viruses that we had discovered and characterized. And you can see that the closest relatives of these viruses, this whole other clade in the tree, is all endogenous viral sequences in different mammalian genomes. So bats and primates and rodents. And so the closest relatives of these snake viruses actually were this type of integrated endogenous viral sequence. And when we looked at those endogenized sequences, we realized that the ancestor of these snake viruses and these endogenized viral elements that are present in mammalian genomes must be really old. And the way you can tell that is that there's this internal node in the tree, sort of that the bottom arrow is pointing to, and that is a common primate ancestor. That's the common ancestor of primates like humans and marmosets. And this, you can infer that this ancestor existed about 40 million years ago. So it's a very old ancestor. And we know that because of different studies that have characterized the long-term evolution of primates using things like fossils. So if that internal node of the tree is 40 million years ago, then this deeper internal node of the tree that's pointed to by the upper arrow, which represents the ancestor of the virus in the primate Eve, is much older than 40 million years ago. It might even be 100 million years old. But nevertheless, these snake viruses, which are currently infecting snakes in Australia today, are only 45% different than these endogenized viral sequences. So they're sort of remarkably similar to these mammalian sequences, and you can use that difference to calculate an estimated evolutionary rate. And you actually arrive at a rate that's much lower than that 10 to the minus 3 for SARS-CoV-2 sequences, more like 10 to the minus 8 substitutions per site per year. So there's this huge discrepancy between short-term and long-term rate estimates. And in fact, others have characterized this for multiple such estimates. And what you can see from this plot, which shows the rate of evolution on a log axis and substitutions per site per year, and on the x-axis is the time scale of measurement. And you can see that the rate decreases exponentially and predictably with the time scale of measurement. And this is actually a pattern that holds true for all different types of viruses. This plot here shows estimates for retroviruses, but for all different types of viruses, this holds true. So what's going on? Why does this happen? Well, one issue is that saturation, which is the reversion of mutations in genome sequences, will cause this to happen. So saturation will cause rate underestimation over long time frames. And the way this works is that if you have a site in a genome that starts as a C and then it mutates to a T, but then goes back to a C, and say this site in the genome kept toggling back and forth between C and T, what would happen is that you'd have a lot of apparent or a lot of actual mutations, but the number of apparent mutations could be zero or very much lower than the number of actual mutations. So this is called saturation. So on the one hand, saturation leads to rate underestimation, but on the other hand, saturation also, in another sense, reflects that sequences are not actually changing, right? So it's sort of analogous to if you were to, run a mile down the road and then turn around and run back a mile. In one sense, you've run 2 miles, but in another sense, you've gone nowhere. And that's what's happening with saturation. So people have proposed that a sort of a conceptual model for what's happening here, which is that the rapid evolution we see for short-term over the short term of viruses corresponds to viruses pacing a small cage, where there's a lot of movement, but they're really constrained in what they can actually do. Another way to talk about this is to say that most sites in virus sequences are probably highly constrained and highly conserved by their so-called host niche. And maybe only a small subset of sites, like say 15%, is open to exploration. But overall, the sequence is constrained by protein-protein interactions, but also other features of the sequence like RNA secondary structure. So what does this mean to you in a practical sense? Well, one thing is that you can't extrapolate short-term rates far into the past. So for instance, if you looked at West Nile virus and Japanese encephalitis virus, these are two flaviviruses. They share about 70% pairwise nucleotide identity. And if you were to extrapolate short-term rates of how fast, say, West Nile virus evolves, which is about half as fast as SARS-CoV-2 over the short term, into the past, you would estimate that their ancestor existed only 40 to 80 years ago. But that's just like obviously too recent of a time estimate. And if you actually sequence West Nile virus or JEV sequences from 40 to 80 years ago, you'd find that they're much closer to the tip of the tree. So there's a problem with extrapolating short-term rates, and you have to be careful when you use tools like BEAST that use short-term rate estimates and about dating these internal nodes. And a better estimate would put that ancestor at like tens or probably hundreds of thousands of years in the past. The other point is that to point out that we've been living with the same viruses for hundreds of millions of years. Here's those viruses again, those snake viruses, and again, I'm highlighting that internal node in the tree that corresponds to 40 million years ago. Well, you can see that the ancestor of the snake viruses as well as the ancestor of all these contemporary viruses, are even older. So for the most part, virus-host relationships are really old and viruses are really well adapted to their hosts. So really understanding this phenomenon has changed my understanding of virus evolution and virus-host relationships. So whereas I kind of used to think that viruses can respond quickly to selective pressures, so viruses are always changing. Now I think that certain parts of virus genomes evolve rapidly in response to short-term pressures, but most parts of virus genomes are highly constrained and highly conserved, probably because they're already really well adapted to their hosts. Another kind of way I used to think was that some viruses have emerged recently, so viruses probably mostly have recent origins, but really for the most part, hosts have been living with the same sets of viruses for millions of years. So I've really enjoyed learning more about the TDRP. It's been sort of spawned by some of our projects, in particular that old RNA work by Lexi. And yeah, that's what I want to tell you about it. So thanks for your attention.